Hello everybody, welcome to this talk with ACROBiosystems. So in today's video, we'll be talking about cell therapy and its emerging landscape in immuno-oncology and the tools and solutions we provide to support the development of the next generation of cell therapies from discovery to commercialization. So before we get started, let's talk about what cell therapies are and what they promise to be. So the premise of cell therapies are to utilize our own natural cells or any sort of living cells to address diseases, most commonly cancer. There are a wide variety of cell therapies that have been introduced, but in general, they can be separated into three classes. Immune cell therapy, which focuses on culturing and manufacturing immune cells, gene-modified cells, which includes that chimeric antigen receptor or CAR T cell therapies, and finally regenerative medicine, which involves culturing and infusing stem cells into these areas of interest. Looking at the current cell therapy pipeline, cell therapy has slowly started to take a hold of the clinical pipeline, especially in the pharmaceutical industry overall. The most predominant cell therapy is CAR T cell therapies, which is gene-edited T cells designed to express a certain specific receptor against certain markers. Other immune cell therapies and regenerative medicines are less predominant, but are also being investigated. However, kind of looking at the overall landscape, what we see is that cell therapies actually have been increasing significantly by over 41% from 2021 to 2022. And we see this trend over and over from the year, and we expect it from 2022 to 2023. Furthermore, what we see is that 58% of the pipeline is actually dominated by CAR T therapies, especially due to the approval of several CAR T therapies by the FDA. And this has really driven a lot more pipelines to be dominated, especially with CAR and CAR-NK, CAR T, and all those kinds of gene-edited immune cells. So overall, what we see is that the cell therapy pipeline is becoming much more saturated with clinical trials. A lot of what used to be in the research phase is now entering into the preclinical phase or to the clinical trials overall. And what we see is that there's over 724 different kind of clinical trials in the China region, and over 493 in the US region. And these are just two of the bigger markets inside of the entire world. So with so many pipelines being filled with CAR T and other gene-edited cell therapies, one of the kind of key question is like, where is that innovation really lying? Or why are there so many different kinds of therapies? So one thing to note is that there are several directions where cell therapy research is heading. So first really lies with that CAR structure. So what we mean by CAR is it's talking about that receptor that's being expressed on the top of the immune cells that's being gene edited. So this really defines what it binds to, what kind of targets, engineering different kinds of domains to help increase that in vivo persistence to make it last longer within the body. You have your cytokine responses as well as improving that overall safety of the cell therapies. So what we're seeing is that a lot of these current cell therapies, such as Yescarta and other ones that have actually been approved by the FDA, is that there are actually very severe side effects to the point where it's pretty much a backup option when all other treatments options have failed. This unfortunately does happen very commonly, especially with cancers. However, what we think, something to note is that cell therapies, yes, are still something to be always a backup option. And generally, it still doesn't really replace any of those frontline therapies, such as chemotherapy, radiotherapy, and others. So in essence, what we see is that cell therapies actually require a significant amount of risk. So doctors are always trying to balance that risk-benefit before proceeding with any sort of cell therapy treatment. On the other hand, what we see is that cell therapies, something that always wants to be considered is the manufacturing. So this is something that most people won't really think about. Like they're always thinking about like, oh, what's innovative, what's new, this next generation cell therapies. But something else to always consider is pretty much manufacturing. So we're talking about how to make these cell therapies. In general, cell therapy manufacturing is extremely slow and it's fraught with complications, right? So it's extremely expensive. Everything is done manually because it's so customized towards an individual. So these manufacturing costs can exceed around $100,000 US dollars, and it becomes even further inflated before it even reaches the patient. So selecting different types of cells to manufacture, optimizing them, or as well as developing a cell therapy that can be universal or used from donor cells are all different kind of key steps towards reducing the cost of cell therapy in general. So in summary, what happens is that cell therapy research direction really comes into four different things. It comes from 1, reducing your costs, 2 is improving that safety, 3 is decreasing the cytokine response, as well as pretty much not having CRS happen within the body. And then finally, you want to prolong that persistence, and that also comes with your cell therapy efficacy. So here at ACROBiosystems, what we do is that we actually offer a very wide array of tools and solutions to help you throughout the drug development process. So it starts all the way from discovery and development, all the way to chemistry, manufacturing controls, otherwise termed acronymed as CMC, which includes manufacturing quality control, as well as preclinical studies and clinical studies. So each step has all of its critical applications, where discovery and development is mostly focused on scFv discovery, CAR receptor design, whereas manufacturing is all about gene editing, cell culture, activation, expansion. And then you have your quality control, which is about therapeutic efficacy, potency, residual detection. And clinical and preclinical studies are always about safety, pharmacokinetics, pharmacodynamics, immunogenicity, and toxicity evaluation. So for each step here at ACROBiosystems, we are always providing a very wide array of solutions necessary for each application, as well as encompassing a wide array of different kinds of proteins, antibodies, kits, and more for pretty much any biopharmaceutical developer. So for cell therapies in particular, our products are really focused around the latter half of cell therapy development. So this is pretty much including cell culturing, quality control, and preclinical and clinical studies. So these are kind of more of the products that we really want to dive more into depth, and I'll pretty much show kind of more featured products. So in the manufacturing stage of CMC, so cell culturing obviously is a very critical part of this. This really shouldn't be any surprise. So depending on the starting material, whether it comes from PBMCs or for stem cells, there are a lot of wide arrays of reagents and growth factors that you can use to influence that cellular growth into the CAR T cells that you use for cell therapy, right? So on the top, what we see here is actually a really quick workflow of how we get from PBMCs, iPSCs, and the products that we use pretty much to help influence its growth in certain directions. So from PBMCs to iPSCs growing to CD3 plus cells, what we see is that you'll be using a lot of isolation beads. Isolation beads in CD4 and CD8, those will pretty much used to isolate out your iPSCs. And then you'll start adding these different kinds of activation factors, whether it's CD3, anti-CD28, as well as serum-free culture media. And then you can able move on and start trying to develop these CAR T cells, which are TA activation beads, as well as the culture media, as well as different kind of cytokine cocktails, depending on what your precursors for your CAR T cells are. This will happen pretty much over culturing for over 5 to 10 days before you're able to come up with your final product. For example, in this one, we're doing anti-CD19 CAR T cells. So throughout each of these steps, you're going to be using a lot of different things. You're going to use cytokines and growth factors. These are all the main components of your cytokine cocktail, as well as your protein conjugated beads. Our brand is called ActiveMax, and this is pretty much what we offer for a lot of these activation beads, as well as different kind of like antibody conjugated activation beads that you use to help activate these cells. And the last one, of course, is your culture media, as well as your basement membrane extracts, pretty much for more specific environments that certain stem cells need in order to develop them properly. So kind of going more into depth about the first product that we have here is cytokines for cell culture. So here what we do is we actually offer 3 different grades of cytokines for cell culture, and this is kind of across the different kinds of use. Because for cell manufacturing, especially cell culturing, you won't only use it just for manufacturing. You'll pretty much use it in every single stage, starting from discovery and development. So for our pharma developers that are doing pretty much just discovery and you're just doing pretty much research use, you can use RUO grade cytokines, which are pretty much all about what we do is that we offer basically just a really basic cell viability verification as well as stability test to ensure that it pretty much meets most of the standards that most of our customers will require for RUO use. After that, after a lot of this discovery and development has been confirmed, the next step really starts moving into your CMC manufacturing. And this is where you're using more higher grades. You have a little bit more higher requirements for your cytokines in general. So this is where you're looking for that sterile, low endotoxin, animal origin free, as well as pretty much something more similar to GMP grade cytokines. So before I kind of go back and kind of elaborate more on PG, then we could talk about our GMP grade ones. So GMP grades cytokines are pretty much the key type of cytokines or key type of growth factors that we'll use before even entering to clinical studies. So this one has the highest quality control standards. It includes 16 different kinds of batch quality control standards to ensure performance consistency as minimize lot-to-lot stability, as well as a lot of different endotoxin controls and detection to ensure that this is safe to be used as a therapeutic that goes into human body. Okay. So GMP stands for Good Manufacturing Practice. It's produced under a very stringent quality management system. And it also pretty much guarantees that your batch production results, your RSF regulatory documentation is all on the up and up. It's all able to be submitted to the FDA for approval. And basically, we're also able to provide this IND support. So one of the big things that what we do offer between PG and GMP is that PG is kind of that precursor before you move into GMP. Because obviously GMP with all the quality controls really adds up to a lot of different costs. PG, on the other hand, it ensures that it has the same level of quality standards without a lot of that extra documentation that really comes into there, as well as some kind of more minor quality controls that aren't 100% necessary until you actually do reach the clinical stage. Obviously, again, so what it sounds like is that pretty much PG is kind of that transition point between RUO and GMP, where you can use something a little bit cheaper, use something more affordable during your preclinical studies, as well as you're trying to kind of ramp up and move into your clinical studies, and then have that general feel for what GMP products are, and then before actually making that transition to GMP. Next product that I want to talk about is pretty much our cell activation beads, which are anti-CD3, CD20 magnetic beads. So before using all these kinds of beads, especially for activation factors, what we notice is that there have been a lot of challenges that have come into, especially with cell culturing and kind of doing cell manufacturing in general. One thing to note is that science and technology is that there's been a lot of challenges. This includes the patient biofluid component diversity. There have been a lot of isolation challenge pretty much because CAR Ts are so specific to specific immune cells. Isolating the correct cells has always been a challenge, and also especially with immune cells that are kind of low non-targeted cell counts, this can always really be a very big challenge. And then the next one is expansion. So activation of these memory T cells, as well as the non-activation of regulatory T cells, is again something that has always been challenging. It's always been done together, especially if you don't use very specific very target-specific magnetic beads. Again, coming up to this, another challenge would be the production process and quality control. This includes scale-up manufacturing, raw material management, and regulatory compliance and standards. Just pretty much adding the anti-CD3 or CD20 antibodies really poses a lot more challenges in removing it from your final therapeutic, as well as pretty much causing a lot of different kind of unwanted side reactions that really can occur in your final therapy. Of course, this all plays into quality control and ensuring that your final therapeutic is always safe for injection. The last challenge is safety, right? So antibodies, viral removal process validation, magnetic beads, obviously with like the radiation treatment, pretty much ensures that it's safe, as well as having that grade A production formulation and filling environment. These are all different challenges that you have to have when manufacturing cell activation or manufacturing cells, and can always be a challenge. So the solutions that we have here is that pretty much these magnetic beads have a lot of different kinds of benefits to it. So the biggest benefit is having these CD3, CD28 antibody cloning and screening for isolation expansion, as well as the virus clearance process validation. Magnetic beads itself pretty much are uniform particle size. They actually simulate APC stimulation, so this really has a better efficacy in comparison just to adding the anti-CD3 antibodies by itself, as well as the surface functional modifications in order to ensure that there are no cross effects or side effects that are unwanted, as well as this irradiation sterilization and process validation that we actually 100% proof that the magnetic beads are actually completely sterile in accordance with GMP standards. And finally, the conjugation aseptic fill finish. This again is something because we don't have to go back manually to detect as well as remove the anti-CD3. Magnetic beads are pretty much something very conveniently done that can be hands-off. So then basically GMP-grade antibody production, sterile conjugation facilities, and expiration of automated filling are all enabled through the use of these magnetic beads and protein conjugated magnetic beads. Kind of talking about more specifically about our activation beads is that we also do offer something called cell-grade pre-conjugated beads. So this also includes our anti-CD3, CD28 beads, as well as other protein conjugated beads, mostly used for activation. But for example, our CD3, CD28 beads, basically what they are, or what makes them cell-grade, is that they're designed to simulate APC cells. So they have an overall diameter of 5.5 micrometers. They integrate cell sorting and activation pretty much to save time, and it's compatible with a lot of various common culture medias for worry-free cell culture. Based upon a lot of the validation that we do, we also find that it preserves more initial and memory T cell phenotypes, which are ideal especially for cell activation and expansion, as well as pretty much residue-free separation and has high safety and stability. So what makes it cell grade is that we do a very significant amount of background validation in order to ensure that these beads are compatible with cell grade and doesn't introduce any other kinds of contaminants that might affect your final therapeutic. Up is our newly launched product line called Active Mag. So this includes our magnetic microbeads. It's very similar to our activation beads, but this one is mostly specially designed for protein conjugated magnetic beads. Again, they're all basically these magnetic beads are coupled with SA biotin for antigen protein conjugation. You can conjugate pretty much any sort of biotin-related protein onto these beads. And they're designed pretty much as a sterile, ultra low endotoxin level cell culture grade beads for you to use in a wide variety of applications. This also includes for CAR positive cell enrichment, CAR ex vivo analysis, as well as activation and expansion, depending on what kind of biotin-related protein that you conjugated onto them. Pretty much here at ACROBiosystems, we're always emphasizing on providing life science tools and solutions to help our scientists kind of travel through that drug development process, starting from the discovery all the way down to the clinical trials as well as to commercialization. So pretty much as a complement to our products as well, we also offer a lot of customized GMP-grade solutions. So this also includes protein customization, custom magnetic beads, closed system solutions, as well as scale up and scale out manufacturing. Big thing here is that all of our previous talks have always been talking about our manufacturing products. But when it comes to production and quality control is actually a very big part of cell therapy manufacturing. And before any kind of product is released, there's a lot of different kinds of quality controls that need to be done. This also includes a lot of things like cell viability, cell counting, cell phenotyping, as well as sterility. So overall, what we see here is that there are actually a lot of different kinds of detection that needs to be done for product release. This includes flow cytometry, residual magnetic bead counting, residual cytokine detection, potency, based upon a certain set of cytokines, vector copies, which is based on PCR, mycoplasma detection, replication complement vector, and cell viability and cell counting. And then kind of moving a little more downstream, we also have the sterility, which is like bacterial, fungal, and endotoxin evaluations. And then in general, what happens is that sometimes we notice that only partial release testing might remain incomplete during the patient cell infusion, right? And this is also a really big issue that can come in, especially in cell therapies. So again, what we see here is that there's actually a lot of different kinds of things that we need to look for, right? It doesn't, not only just for the therapies, but as well as the raw materials that you use for your therapies. So in general, this also comes down to online monitoring. You need real-time monitoring of cell proliferation, quality, cell counting, viability, and phenotype, as well as very key batch release testing. So this includes safety, identification, purity, dosage, as well as potency. So for your therapies, basically, we have a lot of different kind of solutions and as well as a general idea of pretty much how you should be forming your immune cell therapy analysis and quality control solutions. Right. So these are some guidelines that were found that are pretty much available on the FDA. But what we see here is that there's actually a lot of different kind of quality target product profiles, so QTPP. So depending on the route of administration, for cell therapy, that really isn't much of a choice. It's always through IV. The dosage form itself is always a cell suspension containing cryopreservation medium, as well as single dose infusion. Stability needs to be at least 120 minutes from room temperature. Dosage strength is single administration, as well as the quality, so identification, potency, and functional activity. So again, what they do here is they do a lot of scoring. So it comes with the key quality attributes, the efficacy score, as well as the safety score. And these are kind of things that all come down to how they evaluate whether something is okay for lot releasing, as well as releasing testing items for manufacturing. And these are all the different kinds of QC testing that needs to be done. So what we do is that we actually offer a wide range of products for residual detection. So starting off from this is our nucleases, as well as enzyme residue detection kits. So nucleases in general, or this specific kind of enzyme, is designed to cleave any kind of residual nucleic acid that you might have in your final therapeutic. Because a lot of these things are biological in origin, what you'll have is that you'll have a lot of kind of floating DNA from your viral vector, or as well as from your host cell. And this can all affect pretty much the safety or the output of your final therapeutic. So that's why we offer pretty much GMP grade nucleases as well as premium grade, and this is pretty much for you to use throughout your cell manufacturing development process. So another thing that we also offer is that we also have matrigene gel as well as extracellular matrix proteins. So some of you might understand it as these matrigene gels or these extracellular matrix proteins as pretty much as a sort of cell culture medium, especially to help you do stem cell culture as well as organoid culture. But something a little bit more specific is that matrigene gel actually is very useful for assay development as well, where you can use it as a basis for tumor invasion to see the cell migration through the matrigene gel or extracellular matrix proteins, as well as patient xenografting, as well as angiogenesis assays. And these are all different kinds of reagents that you can use for quality control to see how the efficacy of your final therapeutic goes. We also have CAR expression detection solutions. This also comes with fluorescently labeled CAR proteins, anti-idiotypic antibodies, as well as anti-CAR linker antibodies. And again, we do a very significant amount of validation and verification, looking at its purity, its bioactivity, and then kind of going a little bit farther, doing the CAR detection validation using HEC CAR cells, as well as non-specific binding with PBMCs, the bioactivity, and finally, generally, again, with that batch-to-batch consistency, as well as that high stability evaluations. So overall, like kind of moving into the last one, is that we actually also have a lot of these residual detection kits, as I mentioned beforehand. So we have ELISA kits designed to do residual detection, whether you're trying to look for presence of IL-2, IL-4, IL-7, and other different kinds of ones, to make sure that none of these like inflammatory cytokines actually makes it into your final therapeutic and causes unwanted side effects. Another one as well is that we also do residual host cell DNA detection. So this is again double checking that the host cell that you're using, whether it's E. coli, HEC, or CHO cells, actually like none of the DNA that comes from these cells actually makes it back into your final therapeutic, whether it's an antibody or a CAR T cell. And finally, we also have residual enzyme detection. So whether you have DNAs or trace amounts of DNAs, trace amounts of RNAs, or any kind of nucleic acid flow or nucleases that you used previously in the manufacturing step, we want to make sure that those are all filtered out and are actually all removed from your final therapeutic. And these are detection methods for you to ensure that your final therapeutic actually doesn't have any. So kind of being more specific, our cytokine detection kits right now is one of our more comprehensive product lines. So our cytokine detection kit comes in two different forms. We offer singleplex detection. This one's based on ELISA. So this is usually used for detection of more specific cytokines, like based upon your requirements or what kind of cytokines that you use to detect for efficacy, or as well as pretty much an endpoint, qualifying the endpoint efficacy of your therapeutic. We also have multiplex detection. This is based on cytometric bead array, so that's using flow cytometry. Some of the panels that we offer is Th1, Th2 cytokine panels. So this is including 6 different kinds of cytokines that you can monitor simultaneously, and it's very good. It's a much faster way to evaluate cytokines, especially for large amount of samples that you'll see in preclinical and clinical studies in general. That should be pretty much it for most of our featured products here at ACROBiosystems. So first of all, I just want to say thank you so much for listening to this entire video. For those of you who haven't heard of us or heard of ACROBiosystems in general, afterwards I'll also give a really brief introduction about ACROBiosystems and how you can contact us, or if you're really interested in collaborating with us, some different kinds of things that we offer. Okay, so pretty much as a really quick introduction for ACROBiosystems, this company snapshot, is that ACROBiosystems, what we're doing is that we're striving to be a cornerstone enterprise for global biopharma and healthcare industries. So how we plan on achieving this is pretty much by providing different kinds of solutions, different kind of life science tools for pharmaceutical companies or biotech companies to pretty much help them revolutionize how they do research, as well as providing them the basis materials or reagents that they need, starting with proteins and antibodies and a lot of different other kind of biomolecular reagents. So ACROBiosystems was founded in 2010. We're currently at around 750 or more employees, and we have 13 different locations across the globe. In 2021, we actually listed on one of the stock exchanges, and this is pretty much a really big change for us, where we signify this new change of corporate development. Pretty much ACROBiosystems now actually encompasses 4 different kinds of subsidiaries, starting from ACROBiosystems. So ACROBiosystems' pretty much main focus, pretty much all the products that we talked about beforehand, is in the biological reagents, life science tools, and core raw materials. But within the ACROBiosystems group itself, we also have BioCedin, which is focused on industry collaborations, license in, license out, as well as pretty much just ecosystem development. We also have Condensed Capital. This is pretty much based on investment and incubation sort of company, as well as Active Diagnostics. So this is for companion diagnostic and diagnostic technologies. I think you might have seen a really quick snapshot of the products. That would be the ClinMax products that Active Diagnostics has been offering in tandem with ACROBiosystems' other residual detection kits. So overall, again, ACROBiosystems, we offer a very wide array of life science tools and solutions. So our core products and services really starts off with our recombinant proteins. That's pretty much the main part of where we started and founded this company on. Moving into antibodies as well as other kinds of enzymes. But currently we're expanding our product portfolio into kits, cell lines, basement membrane extracts, magnetic beads, MHC complexes, analytical services, regular services like custom development services, pathology, as well as in vitro diagnostics and companion diagnostics. So another thing is that here at ACROBiosystems, we're actually all about quality. So quality of our products is always our top priority. This really comes into our quality management system, which we're finally able to move into GMP manufacturing. But before then, we're also doing 10 different kinds of analytical methods. Our QC process uses 15 different kinds of QC steps before we even release any of our products. And our quality management system has been externally audited at least 4 times by really big companies. We're talking about like Merck, Thermo, and these other kinds of large pharmaceutical companies. Another big thing that we really want to announce is that we're actually expanding, especially into providing this commercial supply for our global CGT customers. So now we're developing a southern base in China for our GMP facility. So it's going to be a GMP dedicated facility with a scalable production capacity as well as GMP clean rooms. One of the key things that we envision this facility to be is to be very consistently regulatory compliant with all the changing regulations throughout the world, especially on cell therapy, as well as providing a really quick base for customers to scale up their GMP requirements, and then we can help them scale up, as well as provide them with the regulatory support documentation that they need in order to move their therapy from pretty much just the research phase, discovery and development, all the way down into the clinical phases. So finally, this really brings us to the last part is that ACROBiosystems, we're always looking for collaboration. So one of the big things beyond quality is actually as well as innovation through partnerships, right? So whether it's for custom services, whether it's through innovation lab or scale-up production, we're always trying to find ways to collaborate with our customers or collaborate with some of companies that operate kind of near within the fields that we're operating in and see if we can work with them to help offer our own customers more innovative solutions or something that can be really useful for them to help develop the product in general. So this really, again, comes into a lot of different kind of directions, whether it's like antibody discovery, design production, recombinant protein expression systems, anything pretty much related to cell and gene therapy, diagnostic kits, neuroscience, as well as high throughput screening. So this could be analytical methods, clinical samples, and pretty much based upon our product catalog, we're actually operating in a lot of different fields. So there's obviously a very wide array of possible innovation collaboration directions that we're always interested in. Yep. So that's pretty much all it is for this really quick introduction for about ACROBiosystems. Again, I just want to thank everybody for listening. And pretty much, if you want to learn more about us, feel free to scan the QR code here. But if anything else, I hope you have a really great day, and thank you for listening.